Lindsay.
Hey Lauren, are you excited for the start of the semester?
Yes, I'm super stoked about BCOR 101, but I'm nervous because my memory is a bit fuzzy after the long summer.
Girl, same. I think if we go over chromosomes, the cell cycle, mitosis, and meiosis before class starts, we'll start off on the right foot.
Great, let's start off reviewing chromosomes. But wait, should we animate our hands first?
Nah, we don't need our hands. Our minds are the most powerful tool here. Let's go! Remember, chromosomes are found in the nucleus of eukaryotic cells. They each contain one long strand of coiled up DNA. This is why we usually look like a mashup of our parents. We inherit chromosomes from each of them.
That's right. And if I'm remembering correctly, I think the tips of the chromosomes are telomeres and the constricted region in the middle is a centromere, right?
Girl, you know it is. Telomeres are at the end of the chromosome and protect it from damage. The centromere is the region of the chromosome that holds two sister chromatids together after replication.
And what about that structure peeking out from behind the centromere?
That's the kinetochore. It's a disc-shaped protein structure on the centromere, and it's where spindle fibers attach during division. That'll be important later when we review meiosis.
Remind me again what a sister chromatid is? Is that why that chromosome looks like it's doubled compared to the last one?
Sister chromatids are two identical copies of a chromosome that are held together at the centromere. Each chromatid consists of a single DNA molecule. We count chromosomes by centromeres though, and we count sister chromatids as DNA molecules. So the first chromosome is the one chromosome with one DNA molecule, and this one is still one chromosome but with two DNA molecules.
Oh right, that rings a bell. And since I always get confused on the difference between sister chromatids and homologous chromosomes, let's look at this karyotype to make it easier. We can visualize the chromosomes with and without their sister chromatids. This first karyotype shows human chromosomes. We have 23 pairs, which makes 46 total. You'll notice these chromosomes do not have sister chromatids, so the number of chromosomes and DNA molecules are the same, 46. In this next karyotype, we can see each chromosome has a sister chromatid. To determine the number of chromosomes, we count those centromeres. So we would still count 46. But how many DNA molecules do you calculate, Lindsay?
Oh man, I wasn't ready to do math today, but I got this. Each of these 46 chromosomes has two DNA molecules, thanks to those sister chromatids, so I'll multiply 46 times 2, carry the 1, and that's 92 DNA molecules.
Perfect! Now, regardless of how many DNA molecules the chromosomes have, both of these karyotypes are showing chromosomes from diploid cells. Most cells in the human body are diploid, which means they each have two copies of chromosomes, or 2N. The N, or the number of chromosome pairs in this case, is
23. We get one set of these 23 chromosomes from mom and another set from dad. These pairs are called homologous chromosomes. And out of these 23 pairs, there are 22 autosomal or numbered chromosome pairs and one pair of sex chromosomes. These can be either XY, the X inherited from mom and the Y inherited from dad, like in this karyotype, or XX, where you inherit one X chromosome from mom and one from dad, which would look something like this.
Exactly. Homologous chromosomes are matched pairs with the same length, centromere location, and genes for the same traits at corresponding loci.
While we're on the subject, I was wondering if we could review more about genes and loci on a chromosome. How does that work with homologous pairs?
Sure thing. So these are a pair of homologous chromosomes, or homologs for short. Remember, a locus is a position on a chromosome where a specific gene is located. Locus, location, easy to remember, right? Let's say here is the locus for MC1R, a gene associated with the presence of red hair. This gene will be found at the same position on each homolog, and each copy of this gene is called an allele. However, two alleles aren't necessarily identical. This red allele is for the presence of red hair, big A, and this yellow allele is for no presence, little a. Together, these alleles are called a genotype. If an allele on a gene locus is different from its homolog counterpart, it has a heterozygous genotype. This genotype would be big A, little a, and because it has differing alleles, it's heterozygous. If we look at another pair of homologous chromosomes, you can see that although the loci are the same, this time the allele on one gene locus is the same as its homologous counterpart. They both do not have the presence of red hair. This would have a genotype of a, and we would call it homozygous.
So if you have a sibling with a different hair color, they most likely inherited a different combination of alleles from your parents. We'll talk more about how siblings can inherit different alleles later on, but I think that's a perfect stopping point until we meet next time. Bye for now!